Welcome to G Fun Facts Online. In laboratories around the world, a new kind of machine is taking its first, tentative steps. These aren't robots of steel and wire, but creations of living muscle and 3D-printed skeletons. This is the revolutionary world of biohybrid robotics, where biology and engineering converge to create machines that blur the very definition of organism and automaton. These biobots, powered by the rhythmic contractions of living cells, promise a future of machines that can adapt, heal, and move with the unparalleled efficiency of nature itself. Today, we're journeying into this fascinating landscape to understand how these living machines are built, controlled, and what they might mean for our future. The dream of artificial life is ancient, but the modern era of muscle-powered robotics is very new. It truly kicked off in 2012, when researchers at the University of Illinois demonstrated the first walking biobots. These tiny 7mm long machines had a simple 3D-printed hydrogel skeleton and were powered by heart muscle cells from rats. It was a groundbreaking achievement, but there was a problem. Heart cells beat constantly and spontaneously. You couldn't tell the bot to stop or start. This limitation led to the next major leap in 2014. The same research group unveiled a new generation of bots powered by skeletal muscle instead. Skeletal muscle is the type that allows us to move voluntarily, and in these bots it could be controlled by electrical pulses. Suddenly, researchers had command over the biobots' movement. They could start them, stop them, and even change their speed by adjusting the frequency of the electrical zaps. This was a monumental shift from simply watching a biological process to controlling it within a machine. Then, in 2016, a team at Harvard University, led by Kevin Kit Parker, created something that captured the world's imagination: a tiny, biohybrid stingray. It had a gold skeleton, a silicone body, and was powered by rat heart muscle cells. But these weren't ordinary cells. They were genetically engineered to contract in response to light. By using different frequencies of light on its fins, researchers could actually steer the robotic ray through a liquid. This, along with other creations like a jellyfish-inspired swimmer, showed how much more sophisticated the control mechanisms were becoming. So how exactly do you build one of these part-living, part-machine marvels? At the heart of every biohybrid robot is a partnership between a biological actuator, which provides the power, and a synthetic scaffold, which provides the structure. The choice of muscle is one of the most important decisions. As we heard, the first bots used cardiomyocytes or heart muscle cells. Their main advantage is that they contract on their own, spontaneously and rhythmically. This means you don't need a constant external trigger to create movement, which simplifies the design. But their greatest strength is also their biggest weakness. Because they beat constantly, it's very difficult to have precise control over the robot. You can't easily tell it to stop, start, or turn on a dime. That's why many researchers have moved on to skeletal muscle tissue. Unlike heart muscle, skeletal muscle cells only contract when they receive an external signal, like an electrical pulse. This gives researchers precise control over the robot's movement, timing, and speed. Skeletal muscle is also much stronger, leading to more powerful and faster biobots. Even more impressively, it has the capacity for self-repair and can be trained through repeated stimulation to become stronger, just like our own muscles get stronger with exercise. The other half of the equation is the scaffold, which acts as the robot's skeleton. This isn't just a passive frame, it's a crucial part of the system. The scaffold is typically created using 3D printing, which allows for complex and customized designs to be made quickly. They're usually built from soft, flexible polymers that can bend and deform when the muscle contracts. The most common materials are hydrogels. Hydrogels are polymers that can hold huge amounts of water, creating a soft, gelatin-like consistency that's very similar to the natural environment of cells in living tissue. Some are natural, like collagen or gelatin, which provide a great surface for muscle cells to attach and grow. Others are synthetic, like a polymer called PEG, which allows researchers to fine-tune the stiffness and elasticity of the scaffold. The design of this scaffold is incredibly clever. Researchers are now building smart scaffolds that actively help the muscle develop. For example, they can 3D print microscopic grooves or patterns onto the surface. As the muscle cells grow, these grooves guide them to line up and fuse into long, organized muscle fibers. This alignment is essential for creating a strong, coordinated contraction. The scaffold can also have integrated features like little posts that act as anchor points for the muscle, similar to how our tendons attach muscle to bone. These same posts can even double as the Biobot's feet. The creation process itself is a delicate art. It begins in a petri dish with muscle precursor cells called myoblasts. These cells are suspended in a nutrient-rich hydrogel, and this mixture is then seeded onto the 3D-printed scaffold. The next step is encouraging these individual cells to differentiate and fuse together into long, powerful muscle fibers. This often involves a training regimen. Just as our muscles grow stronger with exercise, this engineered tissue benefits from stimulation. This can be done by applying low-frequency electrical pulses, which mimics the signals muscles receive from nerves in the body. It helps the muscle fibers mature and align, making them stronger. Once the muscle tissue is fully formed and integrated, the Biobot is ready to be brought to life. The most common way to control skeletal muscle bots is with electrical stimulation. You can place the entire bot between two electrodes and apply an electric field, causing all the muscle to contract at once. Or, for more precise control, you can embed microelectrodes directly into the scaffold near specific muscle groups. This allows you to, for example, activate the muscle on just one leg to make the robot turn. But an even more precise method is called optogenetics. This is a technique borrowed from neuroscience. First, the muscle cells are genetically engineered to produce a light-sensitive protein. This protein acts like a tiny, light-activated switch. When you shine a specific wavelength of light on the cell, the switch flips, and the muscle contracts. This is non-invasive and gives researchers incredible control. By shining a focused beam of light, they can activate very specific regions of muscle, allowing for fine-tuned steering. The ultimate goal, however, is to replicate the body's own control system, the neuromuscular junction. This is the connection where a motor neuron sends a signal to a muscle fiber. Researchers are now co-culturing motor neurons, along with the muscle tissue on the scaffold. These spinobots have been shown to produce a more natural, coordinated walking gait. It's a critical step toward creating truly autonomous biobots that can sense their environment and decide how to move. The results of all this work are a true menagerie of strange and wonderful creations. The first walkers were simple, asymmetrical designs with a long, flexible leg that pulsed to propel the bot forward. Now we have two-legged bots that can be steered by light, and even spinobots with integrated spinal cord tissue for a more natural rhythm. The principles have also been applied to create swimmers. There's the medusoid, which mimics the pumping motion of a jellyfish's bell. And of course, there's the famous light-guided stingray. In a fascinating twist, some researchers are augmenting living animals. By implanting a microelectronic pacemaker into live jellyfish, scientists made them swim 3 times faster than normal. These cyborg jellyfish could one day be used for large-scale ocean monitoring, collecting data on temperature and salinity. Beyond just moving around, this technology is being used for manipulation. Researchers have created soft robotic grippers, powered by muscle, that can gently grasp and lift delicate objects. Imagine a biohybrid hand with multi-jointed fingers, each controlled by a separate muscle. These could have future applications in microsurgery, where a gentle but precise touch is critical. The potential applications are vast and sound like science fiction. In medicine, tiny biobots could one day navigate the human body to deliver drugs directly to cancer cells or perform minimally invasive surgery. Because they're made of biological materials, they could be highly biocompatible, reducing the risk of an immune response. They could also be used for environmental monitoring, with biodegradable bots released into the ocean to clean up oil spills. And they serve as incredible platforms for scientific research, allowing us to study muscle function and nerve integration in a controlled, engineered environment. However, the field is still young and faces enormous challenges. Most biobots are still tiny, on the millimeter or centimeter scale. Scaling them up is a huge hurdle, mainly because of the nutrient problem. A large piece of muscle tissue needs a blood supply to deliver nutrients and remove waste. Diffusion only works over very short distances. To build bigger bots, we'll need to engineer a built-in vascular network, a tiny circulatory system, which is a feat of tissue engineering we haven't yet perfected. There are also issues with longevity. The living tissue is fragile and requires a perfectly controlled lab environment to survive. Their speed and strength are still low compared to traditional robots, and the muscles can get fatigued. And creating true autonomy, where a bot can sense its world and make its own decisions, will require integrating complex neural circuits, which is still a long way off. But perhaps the most profound challenges are ethical. As we blur the line between living organism and machine, we face deep questions. What is the moral status of a biohybrid creation? If we integrate complex neural tissues, could these bots develop some form of sentience, the capacity to feel pleasure or pain? If a biobot can feel, does it have rights? Then there's the issue of animal welfare, since many current bots use cells from animals. There's a strong push to move towards using human-derived stem cell lines, which are a more renewable and less ethically fraught source. These are not easy questions, and they require a deep, ongoing dialogue between scientists, ethicists, and the public. The journey into biohybrid robotics is just beginning. The challenges are formidable, but the pace of innovation is staggering. The future of medicine could be populated by microscopic, muscle-powered surgeons. Amputees might one day have biohybrid limbs seamlessly integrated with their own nervous system. Our oceans could be protected by fleets of self-powered, biodegradable bots. The creation of these living machines forces us to confront what it means to be alive and what our responsibilities are as creators. The path forward requires not just scientific genius, but careful ethical thought and a commitment to responsible innovation. One thing is certain: the future of robotics will be softer, smarter, and more alive than we ever thought possible.